Hello, Gnanostones. We should be live now. I hope. I had a few technical issues. The most important part of this decided to stop working, which is the microscope camera. But it should be working now, and people can hear me. How is the sound? I'm guessing it's probably echoey. But that's kind of how it has to be. Everything is pushed to the side. I'm a really weird setup, and I will probably hit the camera and stuff. This is not gonna be professional at all. This is just me fucking around. Okay, so I have this. This is a camera that is. This is a microscope that the camera is not focusing on. And I'm gonna move away from the mic, and we will see how this goes. And let's see if we can zoom in a bit. So, this is my microscope. I got it 2 years ago, but this thing is probably at least 70 years old. It was made in the sometimes around the 1940s. 46 is. I should try that again. So, this thing is a Reichard. I'm probably mispronouncing that, which was produced in Vienna, so the capital of Austria. And I don't know if you can see. There's some writing on. There's some writing on it right here. That's the serial number. It's number 200 something thousand, 200,000 something. I found, obviously after I got this, I went online and started looking, for other things, for other microscopes to get an idea of how old this was. I'll get to the story of how I got this in a bit. But I found other microscopes of different models. And assuming that Reichard used their, they didn't use different serial numbers for different models, which I don't think, because in that case they would have produced a lot of these, like a lot. That one was started with 2.3. Mine starts with 2.2, so mine must be older than that. And that one was estimated to be from the 1940s. It actually comes with a rather beautiful case in wood. This thing right here. That's not very handy. Hold on. I just pulled my lamp off. That's stupid. There we go. So, this is the case it comes in. As you can see, this isn't just like a random case someone put it in. This was specifically made for it. You can see how it fits in here. And I actually know the model number of it, thanks to. I need to open that. I forgot to open something in my confusion over trying to get this to work. So that's great. Let me find it. Here we go. Sorry for the silence. I'm trying to find something. This should work. Here we go. So, I went online, as I said, and started looking for things. And one of the things I found was an old catalog for Reichard. And it actually has my microscope here. Right here. As you can see, it's a monocular, which basically means that it has one hole to look in and not two. laboratory microscope RC. So RC is the model with a substage lamp Lux TB. So that's the lamp underneath. As you can see up here on this model, sorry, what was really common at the time was to use a mirror and just use the ambient light to shine through. And you can actually, I found other people selling the same model as I have, but with the with a mirror instead, and you can actually take the thing off. As I said before, I've had this for 2 years. The reason it has taken me 2 years to actually get around to doing anything was that I didn't know how to change the bulb. I later figured that out, or rather my dad did. Turns out you just have to be violent with it. I was too afraid of breaking it. But the old bulb, which was the original bulb, I assume, or at least like an old incandescent bulb, got so hot that I would burn myself on it, which is not ideal. Also, anything you put on it would evaporate. The water would evaporate like in 2 minutes, which is also not ideal. And by the time I figured out what to actually do, how to change the bulb and get a new bulb in it, it was too late to actually show anything, because it was fall and winter at that point. So yes, violence is always the answer. Okay, so before we start actually looking at it, I want to tell the story of this microscope. Or at least as much of it as I know. As I said, I got this 2 years ago from an old man, a pensioner, who, and this is what he told me, he used to work for a subsidiary, not a subsidiary, but for a distribution company in Denmark that distributed the microscopes for Reichard, which I actually have a picture of. There we go. So that's not it. This is what this is on the inside of the door of the case at the top. What you see at the top, I'll get back to that. But what you see at the bottom, that's the name of the distributor. That's and it's so old that chemical, the Danish word for chemical, is spelled with a ch. That in modern Danish that would be spelled with a k. I'm not sure. It's a bit like, I'm assuming this company started out producing chemicals, and then later became a distributor of microscopes, because the people that bought this, that had this originally, wasn't very chemically. As you can see at the top here, it's the top one. It says Den Kongelige Veterinær og Landbrugshøjskole, that which translates roughly as the Royal Veterinary and Agricultural University, which was an old thing here in Copenhagen, where all the vets and all the like people who work with farming and farm animal and agricultural stuff, other than obviously the farmers, would be educated. It still exists, but now it's just a part of the University of Copenhagen. And underneath it says Plantefysiologisk Lab, so that's plant physiological laboratory. So this was originally used in that laboratory for presumably looking at plants, which is a bit annoying, because plants don't tend to move a lot, and they're pretty easy to keep track of, which means my microscope doesn't have this, which is a mechanical stage, which is to move things around very finely. So I'll have to use my fingers. So once we actually start looking at things, it's gonna be kind of jerky. Also, keep in mind that we will be looking through 70-year-old optics, but they'll still work surprisingly well. Like, I mean, glass doesn't rot, so it's to be expected, and it's probably been sitting in that case for a long time. But it will also go through both the camera and then YouTube. So back to the story. So the guy I bought it from, sorry, I'm reading the chat. That's why I go quiet. The guy I bought it from, this pensioner, he worked at this distributor, and then at some point, like, he probably didn't work there when they actually bought it, because he was in like his late 70s, and I don't think he worked there as a 10-year-old. But at some point, the Royal Veterinarian and Agricultural University obviously got new microscopes and didn't need these anymore. So they returned them. Apparently that was common. I didn't know that you could just return to the distributor, which at that point, Reichard had at that point, or either Reichard, the guy wasn't very clear, he was old, so that's to be expected. In any case, he still worked at probably the distributor, who then got it back. And they, like, the distributor obviously didn't have anything to use this for. It's an old microscope. They probably had better. They probably had a lot better microscope at that point. So they were probably just gonna throw them away, or they were going to, or they gave the offer to the workers to get one. So this guy said, "Oh yeah, sure, I will take that." So he got one and went home and probably used it. I don't know. And then he probably has had this sitting for like 30 years. And then I bought it, which is kind of cool. So I have this really old thing that still works. Like, the only thing I had to fix was to use, other than changing bulb, which isn't technically a fix. The only thing I had to do to it to get it to work was to use a bit of violence on the focus knob, on the fine focus knob, to get it to work. So, but enough of this. Let's move on to the front here. So, I showed this picture before where it says PL18, and also says that on the microscope itself. In fact, it also says it on the objectives. I'm not sure what that means. I'm assuming that PL stands for plant physiological laboratory, but it's also on the microscope itself. So either this is like it's because it's not actually an RC, but it's like a special model that the university ordered or something like that, or that's what the model was called in Denmark. I don't know. So that's a bit of a mystery. Anyways, let's actually get to the fun part. So, let's, here, I'll take this, this goes over here. So I have a few things we can look at, some of them biological, well, most of them biological, actually all but one is biological, but I also have my phone, and on my phone I have a completely yellow picture, because hopefully a moment we can show something really, really cool about eyes. So let's start with that. There we go. Let's see. Let's go up. Now the question is if we can actually see anything here. So what you're looking at here is the pixels. There we go. Let's switch to this. So these are the pixels at high magnification. Now remember, the picture I put on there is completely yellow. It looks a bit greenish in the light here, but trust me, it's completely yellow. So what's going on here? Because there's no yellow light here. All there is red and green light. Oh, and also, yeah, there's a bit of an optical illusion. The reason for this is the way your brain sees yellow light, and this is true for this is true for almost all screens. There are a few screens that have yellow subpixels. Yep, Tornado Pod is correct. They the red and green pixels combine to make yellow light, but it's not yellow light like the yellow light that comes out of a yellow light bulb. Instead, what happens is this. So, inside your eye, in the back of it, on your retina, you have four. Technically, you have three different light-sensitive cells, but you only use two of the kinds to see with. The last one is for things like your pupil and adjusting to the brightness. So you have the ones you actually see with are rods and cones, and there are three types of cones. Rods are what you used to see when it's dark. They don't see color. They just see black and white. But the cones, you have three different types of cones. As it says here, you have S, M, and L. Well, which corresponds to blue, green, and red. But as you notice, there's no yellow. So the way your brain sees yellow is actually that it activates yellow light would fall somewhere in the middle between the red and green. Like obviously that's how the spectrum looks if you like look up an electromagnetic, the visual part of the electromagnetic spectrum. And as you can see, like they have peaks where they're most sensitive, where they absorb the most of wave. That's the wavelength where they absorb the most light. But in the middle here, they still absorb something. So the way your brain actually detects yellow light is by seeing that both the red and the green get activated. And obviously, you could, like with normal yellow light, they just both get activated because they both absorb yellow light. But you can trick the brain into thinking that that's happening by just activating both of them using both red and green subpixels. So that's how your phone is able to create yellow stuff. Create yellow stuff on the screen without actually producing any yellow light. So that's the technical thing here. That's the non-biological. But I thought it's cool, so I wanted to show you. Okay, let's see. I have a few different things we can look at. I have something some tissue that came with it. That's possibly human, if you want to see that. I also have some yeast. I have one of my hairs. Head hair, not any other kind of hair. Although, yes, my leg hair is long enough that I could take that. And then I also have some slices of onion. We can hopefully see something with there. I actually did think about doing that, showing sperm cells, but I kind of decided against it on the grounds that that's a bit weird. I had the thought, but the thought quickly disappeared again. And then I also have some moss, and I have some lake water that I went out at and got. Okay, I have to point out here to keep the illusion up. The actual blob isn't here today. I'm the assistant. So I have hit here. So, what do you want to start with, Chad? Okay, this just turned into sperm samples. Oh, Creation House. Sadly, my skin isn't really that dry, so I couldn't really get anything off of me. So I would show some dead skin cells. That would be fun, but I couldn't get any. Well, I don't have a sperm sample, and I don't think you wanna wait for me to get one. That sounded weird. So instead, let's do this. Okay. This probably isn't showing very well on it. There is some writing up here. It just says 1, 2, 4. It just says numbers. I have no idea what it actually is. But this came with the case somehow, like, it's a bit weird that this came with the case with the microscope, because obviously this was originally used in a plant physiology laboratory. So why is there a piece of animal tissue? So that's a bit weird, but I'm guessing it probably got used for something else later on. Anytime I go quiet and just laugh to myself, it's because I'm looking at the chat. I should probably let's close the chat for a moment here, minimize that so I can actually do stuff. So let's put this in and then turn this on and switch over to this. So, I didn't mention that the magnification levels we have here is 6.3, 20, and 45. Now, if you ask me, like, you can go ahead and ask me what magnification we're actually seeing this things under. But that depends on how big your screen is, because obviously when you take when the camera detects it, the larger your screen is, the larger the picture is gonna be. So that also means that it's technically been magnified more. And also when you have the camera, it also depends on where exactly the sensor is compared to the focal point of the light coming up through the. So the thing did come with a couple of oculars. That's the fancy name for the eyepiece pieces. There's a 12 and a 6. This is 6 one. And the maximum I can do there is obviously 12 times 45. It's like, because that's how you do a magnification. You just multiply the objective, which is the bottom part here, with the magnification of the eyepiece, and the maximum is gonna be 12 times 45, which is 540, which is actually quite high for this kind of microscope. Normally microscopes would go up, like if you went out and bought a laboratory microscope today, you would probably get, the maximum would be 400 times magnification. The reason for that is that when you go higher, technically, it's physically possible to magnify using a light microscope to magnify up to about 1,500 times magnification. But if you go higher than that, what happens is that the magnification becomes too small compared to the wavelength of the light you're looking at. So you just can't see anything anymore. You can't magnify it anymore. But in order to go higher than about 500, you need oil immersion. That has to do with the fact, like, instead of using water, you have to use oil, because oil has about the same refractive index as glass. You can actually test this. If you get like a cup and a smaller cup and fill it up with cooking oil, obviously they don't use cooking oil in the microscope, but it still works, and then dump the smaller glass into the bigger glass, it will just look like it disappears because it's the same refractive index. So the light doesn't, like, there's no change in light, so there's no change in how the light behaves when moving from the oil to the glass. So it just ends up disappearing. It looks really, really cool, actually. So you need to do that to get higher. Okay, this is on the 6.3. And yes, that is a weird number. I'm not quite sure why it's on that. Let's see what we can find here. So obviously this is just like small bits and pieces. I'm not even sure if I'm actually on the... There, it looks like there's something here. There we go. Look at that. This is weird because it's reversed. So this is intestine, and I will show you how I know that in a bit. This is a really bad piece. We need to find, like, it's been cut. The way, if you could actually look at it, it's obviously like a glass plate with a that is on, and then it's like a series of cuts through this tissue. So it's like small, like, crescent moon shapes in a row. There's something. Oh, ooh, that's a good one. Well, I'm not entirely sure if it's human. It could also be mice, mouse intestine or cow intestine. I really don't know. I don't really have a way to know that, because at this level it pretty much looks the same, and this is probably a lot of, like, decades old. So I'm not, like, it's unclear whether or not, like, I don't even think I would be able to identify it, because well, this is a good one, because we can actually see the the cool thing here. Let's turn around and go to 20. And obviously now I need to adjust the focus. Oh, I hit it perfectly. So what you're looking at here... No, there is... oh, okay, you meant the human thing as a joke. Well, no, there is a chance that there's a human tissue. I don't know. It came with the box, obviously. So what you're seeing here... Actually, let's see if we can... That looks fine. What you're seeing here is small intestine, and I know that because what you're looking at is what's called a villi. So inside your intestine, your small intestine, where the majority of the food is absorbed, you obviously need as large of a surface area as possible to get the, like, because the more surface area you have, the more you can transport it from the inside of the intestine and into the blood. So what your intestines have is these little like, they look like little, like it looks like a carpet almost with long fat hairs, like fat threads sticking out, or like a brush. It's actually called the surface of it is actually called the brush border, because on each of these villi there is something called microvilli. So you can see the darker areas on the pictures. Those are the surface of the cells that would have phased into the middle of the intestine. They've probably been colored with something, stained with something, possibly some kind of immunothing where you use antibodies with some kind of dye on it or something, because it's clear that they've stained it in a way where the brush border is stained, or something in that area is stained. That surface of the cell membrane. But these, like the inside of like the surface, in the inside surface of each of the villi, the inside of each of the villi, the surface of them has what's known as microvilli, which is like smaller, like they look like villi, just much smaller, and they're basically the same, but instead of being the surface of the intestine, where it's a bunch of individual cells, it's the actual cell membrane that does it. So it increases the surface area even more. The cool thing here, you can see, if we try and adjust the focus a bit, can you see there's like something in the middle? Like you have the bluish purple border, which is the surface of the villi, and then you have a pink layer, that's the cell that was attached to. But then you have an area in the middle. That's the capillaries. That's the blood vessels that runs into each villi has blood vessels running into it, and then out again, that sucks up all the nutrients that the cells have in your intestines have absorbed, goes into and gets out in the rest of your body. So that's also why I know how I know what this is, because I can identify this area, because it's a very characteristic structure. So that's a bit of Dark Babylon. There is a bit of fractal to it in some sense. So that's something that came with it, which is, I think it's like, Creation House, I'm not sure why this is making you hungry. I mean, maybe you're thinking of sausage, which is made from intestines. I'm not sure. So that's this thing. We could try to zoom in further, but I actually think we see more of it here, more of the detail. Yeah, don't let Nassim Haramein know about this. That would be bad. So that's the intestines. What else do we have? Let's go with my hair. So what I did here is here. It's really hard to see. Okay. What I did here is I pulled out a hair and I super glued it to the glass so it doesn't fly away, and then put 2 cover slips. That's the glass you put over. So you have a slide and then you have a cover slip. Put it over so it actually sticks. Otherwise it's just a small drop, because you need pressure to activate super glue. And my hair should be in here somewhere. Let's switch over to this and see if we can find something. There we go. Look at that. Let's go to some more magnification. It seems to be slightly moving, because but that's to be expected since the middle of the hair that we're actually looking at isn't secured, so that can obviously still move a bit. Like you can see there's a bunch of dirt and shit on it, but you can actually tell. Oh, here we go. Can you see the scale? Can you see how it has like it has like wrinkles on it? It almost looks a bit like a brain or something like that, or like a palm tree. Those are the well, not literal shit, but like stuff. Oh, okay. Let's see what I mean. That it's a bit jerky when I try to move it around because I don't have a micro manipulator. I would love to get one, but the problem is it's really hard to find because this is so old. It's really hard to look up the specifications of it. But what you're seeing here is the you can see the surface of the hair. Now there isn't a lot of color here, and that's because I'm blonde. So my hair is essentially colorless. Well, it's not white, but there's very little pigmentation in it. So that's also why we yeah, it probably would be cheaper to buy a modern microscope at this point, unless the measurements haven't changed and I can find an old I don't know, sorry, I can find a new one. I can find a new one that still fits in it, like, or find a new one that I can make to fit in it. Like it's just like a few holes that things have to go in. That sounded wrong. But the thing you're seeing, making a micro manipulator thoughts and memory is kind of hard because it has to be really precise, because it obviously has to move like fractions of a millimeter every time like you turn one of the knobs, and that's really hard to make. Like it has to be really precise. But the scales you're seeing, the lines you're seeing here are actually the scales. Hair actually looks a lot like, has the cells, the dead cells, the former cells in your hair and in your nails, because they're made out of the same thing. They're made out of the same kind of cells as you have in the top layer of your skin. They've been keratinized, meaning that they're basically like they're dead. You take a cell and then it slowly dies, and as it dies, what it does is that it will produce a bunch of keratin. That's the part of your skin that makes it waterproof and like makes it so it like functions as skin. When you get a scratch where you start to like if you fall and get and scrape yourself, what you do is you scrape that dead layer of skin. It's called the epidermis. You scrape that off and then expose the living tissue underneath. And hair is essentially like they're made out of keratinized cell too. They're made out of keratinized cell too, but they're like stacked together in these scales. So each of these scales is a cell that's sort of on the outside. So your hair isn't actually isn't smooth. Like it's not just like a line. It actually has these little these little scaly bits on it, which is also why like something like if, like lice can actually crawl on it because there's something to hang on of. To hang on to. This works a lot better than I expected actually. Oh, I think I saw quickie. I can't show the hair root because I didn't get the hair root out with it. I didn't pull hard enough. And frankly, that would just be a big blob of like dark because they're rather big. So actually looking through it. Actually looking through it is kind of hard. I need to stop reading the chat as much. I'm sorry, chat. I get distracted by it. So that's the that's my hair. I think that's kind of neat. What else can we look at? Let's go with another of the slides I've pre-prepared, and go with this, which is some onion. Hopefully these will be thick enough that I can actually, thin enough that I can actually look through them. They're not, they're too thick for to actually fit in here, which is kind of a problem. Let's see what we can find. Hey! Look at that! So there's obviously some bubbles in the way. Yeah, smelly is correct. You can actually feel the scales if you pull a long hair out, take a long hair out, and like pull it, your nails across it. You can actually feel the resistance from them. So there's obviously quite a few bubbles here because this is a fresh onion that I just put a piece of glass over. But you can actually see the cells underneath. And you can see that they're also kind of bubble-like. Those are plant cells. And what you're seeing here is the cellulose that's keeping, because all plant cells, unlike animal cells, have a cell wall made out of cellulose. So the same thing one of the things my table is made out of. There's also some other things in here. The fun thing about onions is that a lot of people think they are that they're roots. They're actually not. They're actually modified leaves. So each layer of an onion is actually a leaf, where that stores a bunch of like starches and other sugars. So it's sort of the winter lunchbox. Not the bubbles, not the bubbles with the black thing around them. That's really like the really black outline. Those are actual like air bubbles. But the other things down here, the much like thinner ones where you can actually see detail, those are the actual cells of the onion. I don't know, Gio. I've never actually looked. I've never had the chance to look at ogre intestines under a microscope, or look at an ogre in general. So if you can find me one, then I'll be happy to kill it and dissect it. Because fuck ogres. I guess I'm apparently anti-ogre. Yes, I'm very against this fictional creature that doesn't exist. Yeah, Yellow, that is correct. This is why we stain cells, but you can still see something. Actually, Oh, you can really see the network here of cellulose. Like you can see all the ridges in it. That's pretty cool. Recent vision, yes, I am making my normal videos. I have one in the works. I'm almost done with the audio. It should be out in like the next 2 weeks. So you can actually see all the cellulose. And obviously, like the way onion, like if you leave an onion in the ground, it will stay there. Like it's the plant's like winter lunch. It's rations to keep it through the winter when it dies on the top. Where the plant, part of the plant that's above ground dies, and then it stays in the onion stays in the ground. The bulb, the flower bulb stays in there. And then it uses that energy to get out of the ground next year. So, we're running through this pretty fast, but now these are all the things that I like prepared. I prepared. So I keep reading the chat to see what happens, and I get distracted. What we've been looking at, the 3 things here. I've already prepared the plates with prepared the slides, the glass plates for us, because they're a bit easy to prepare. But the 3 last ones I have, we need to actually do a bit of work. So let's remove this and put it over here. Over here being the table behind me. Well, an assistant should be nice, but this is... I mean, I'm already the assistant to the blob, so what can I do? Okay. So, that's the slides and some cover slips. And this. Look at it. Isn't that yummy? Anyone want to take a guess what this one is? I think I actually already told all the things I have, so it's a bit of a cheat. Well, not a cheat. Okay. Yes, Gio is correct. 21% sperm sample. You have a fucking problem. Go see a doctor. It's not supposed to have that color. And we need a pipette. Pipette. It's from an arts and crafts store. It would be nice to have it would be nice to have a proper like pipette, but those are expensive, and I actually don't need it. So, sorry, got distracted again by the chat. I should really stop reading it. So, what we need to do, better do this so guys can actually see what I'm doing. Otherwise, this is just me talking about stuff you can't see. That's kind of stupid. We have this piece of glass, and we need a drop of yeast. This is just like normal baker's yeast. Or, well, to give its actual name, this is spotting yeast because of the way it reproduces. I can get back into that in a bit. Drop of that. And let's see. Just make sure I don't have 2 here. And then the trick here is to like, you obviously can't see this because it's too far away and my hands are in the way, but the trick here is to not get any bubbles under it. That looks good. Now we have a little yeast colored square here. I got something underneath. Fuck it. It wasn't completely perfect. So, let's see what you're saying. Well, Red's Rhetoric can go live in 30 minutes if he wants to. Like, he knew this was coming. Let's see here. Probably go down, that would probably help. Starting to think this might be too opaque. I don't think I actually think I made this, oh, there we go, oh. Look at that, there we are. Yeah. Let's see if we can find this. There we are, look at them. They don't look like much here. And well, the thing is, first of all, yeast is not motile. They don't move. They just sit around. They just sit around, so we won't actually see them do anything here. And they're also very, very small. Well, they're not dead. Like, they're clearly actually alive. They are alive. They're probably not doing a whole lot because I haven't actually given them any food. I could have given them some sugar and stuff, but you wouldn't really see it. Anything. We can try and go up to the 45 times magnification and see what we can see here. See with that. But obviously, as we go, further and further up in magnification, we also get less and less light in there, so the picture gets darker and darker. Now the question is if we can actually see anything at all here. I think they may be too small to actually see. Or at least for me to find them. The problem is I have no idea where I'm actually zoomed in. Not zoomed in, where I'm actually looking. Or if I'm looking at anything that's actually there. There. Oh, that's a much better. Look at them. Haha. I think what we zoomed in were focused on before were probably some dirt on the condenser lens. That's the lens that sits underneath, or possibly some dirt on the thing. Some dirt on the lens inside the objective. But these looks much better. Let's zoom into the whoa! Look at that. Okay, so you can actually see there are some little bacteria and stuff in between them. The movement you see here is not because of the yeast itself. The yeast can't actually move. Well, Jared, the reason I can't keep my tools clean, I actually tried. The objectives have been on the microscope for so long that I can't. They won't come off, and I'm not gonna try and use violence this time. To get them off, and as you can see, it works perfectly fine. There is some bit of some Brownian motion here. There's also the fact that like this, if I knock on the table, you can see that there's movement. I think there are actually bacteria or something in between them. Because obviously, like I just took a glass and put like water and yeast in it. It's not sterile, so like we're gonna get other things. Let's see if there's any of them that they're actually doing any. See, there's multiple layers of them, and they're floating around. So I don't think, since I haven't given them any food, there's none of them that are actually reproducing. No, tornado pelt. Yeast cells are not prokaryotic; they're eukaryotic. Yeast is a type of fungus. Yeah, they're fungi, as Reason Vision says. The reason you don't see any nuclei in them is because you actually don't see like nuclei really. The only time you really see nuclei is when the chromosomes are condensed. When the chromosomes inside is condensed, when they're getting ready to be duplicated in mitosis. So the reason I said before that these are called, like the technical term for these are budding yeast, is that the way they reproduce is that, so a normal cell, like your cells, when they reproduce, you get two cells that are like roughly the same size. No, Reason Vision is actually during mitosis and meiosis, you actually do get it gets condensed down, because the chromosomes, after they've been duplicated, well, kind of during they're being duplicated, it's a bit tricky. Once they've duplicated it, they need to spread it out. Like each new cell needs to get some chromosomes. So and in order to like get them out and get them separated, they're condensed down, and then they're like these weird spindly things, like long chains of proteins that sort of grab onto them and then pull them out. Yes, and smell like E minor is correct. That also happens in meiosis. It's basically anything. It's basically any kind of cell division, and it's correct that you can't. As Yellis says, it's correct that they can't really be read while they're condensed. And as Dark Babylon points out, that yeah, most of the times they're actually like a tangled mess. Your body actually uses quite a lot of energy and time and energy to keep your DNA sort of spread out and not like entangled in the correct way. But if they're condensed, they're not unraveled, and you shouldn't see it, right? Yeah, that's the reason why you don't see it. You don't see it. You don't see it right now. That like they're just like the DNA is in there somewhere, but the DNA is in there somewhere, and they do have a nucleus, but we can't see it because it's not condensed. And yeah, this is pre-beer. Except this is not the kind of yeast you would use to brew beer. So, Oh, did I say the opposite? Okay, let me just so it's completely clear. When the cells are dividing, they condense their DNA, their chromosomes, and they have like the shape you typically associate with chromosomes. The rest of the time, when they're not dividing, when they're just doing their thing, it's not condensed, and it's just like a tangled mess. And well, it's a mess and it's tangled, but it's tangled and messy in an orderly way. Like the different parts are actually like in specific places in relation to each other, because that's part of how the genes are regulated. But so back to what I'm really talking about, the way these reproduce, because when your cells split up, they undergo mitosis and become one. The two are some sort of roughly equal size. Like the cell pretty much just grows bigger and then splits into two. When these little buggers do that, become more, they don't do that. They make a little like a bud. Hence why they're called budding yeast. They're like a little prolapsed thing on the outside. On the outside of them, a little bud, which then the copy of the DNA then goes into, and then that bud goes off, and it's much smaller. Smaller than the other cell, which is kind of weird, but that's how these that's why they're called budding yeast, because that's how they reproduce. And they so we can't actually see if we had if we actually like, I think you can actually see their nuclei if you look really closely. You can sort of see that there is an area inside them. Yeah, you can actually see the nuclei. That's really cool. And as I said, the motion here is just because the water is moving around, because it's being heated up. And you can also see that they're different sizes. I'm just like, yeah, it's kind of trippy to look at. Actually, it would be a neat screensaver. But these are like, they're super important. This species of yeast, both in cancer research, like a lot of what we know about the way the cell cycle, which is the thing that controls when a cell should divide, and all the processes that goes into dividing, they're controlled by a bunch of different genes and proteins. And when they I lost my thread there, sorry. And when they and like, obviously it's a lot easier to work with yeast cells than to work with human cells, because yeast is supposed to live on their own. Also, you have each of your cells, or most of your cells, have two chromosomes. They come in pairs. But that's called being diploid. But yeast is in this stage is haploid, so it only has one version of it, which means that if you make a mutation in a gene to figure out what it's doing, like an easy way to figure out what a gene is doing is like destroy it and see what goes wrong, and that is one of the techniques that's used in researching what different genes do, what the proteins produced by them do. And that's obviously a lot easier when you it's obviously a lot easier to do when you only have one, because if you had two, then there's a good chance that the other the other, the like the corresponding gene, the other version of the gene that's on the other chromosome would just do the job for the one you destroyed. So you have to kind of get lucky and destroy both, or destroy one of them in such a way that both of them don't work. That's a lot easy. That's like, if you can avoid that and just have this when it's, like, when it's only one, then that's a lot easier. But yeah, they're actually used, like, a lot of, we know about the cell cycle. We know from mutating yeast. And obviously they've also been very huge culturally important. Like in both in brewing and in bread making. Like, they've been very, like, super culturally important in those regards. Look at them go. It's like watching something like a migration of buffalo, even though they're not actually migrating, they're just like flowing. Yeah, tomato pulp. Pretty much, you learn about how things. One of the ways to learn about how things work is to break the gene that makes the protein you want to study, and then you see what happens. Obviously, if it's something where, like, if it's a gene that's really, really crucial, and you, like, where if you destroy it, the cell dies, that doesn't really work. But what you then do is that you have something called oh, I can't remember the word, but you have some mutants where, under like certain circumstances, like for example, like temperature is a really popular one, where at like 20 degrees the protein works pretty much normally, but then when you turn the heat up to like the human body temperature, for example, then it stops working. So you can sort of have them alive and grow them and get a bunch of them, because obviously when you make a mutation in the yeast cell, you only make a mutation in like one cell, and you then have to get that cell to grow and multiply so you can actually have something to work with. And if you have these conditional mutants, that's what they're called. If you have those, then you can avoid that. Like, you can just like have them and then grow them up, yeah, on the 20 degrees, and then put them into the warmer environment and then see what happens. So yeah, I can keep going. Yeast are really, really fun. They're really cool animals. No, they're fungi. But we also have some other things to look at. That's actually like the main thing I wanted to look at wasn't this. The things we've been looking at. I actually went out and got some moss and some ocean water. Ocean water. Wow. Got some lake water. So for anyone who hasn't watched me on a stream before, this is how I normally talk. I'm usually not nearly as coherent and well spoken as I am in the videos. Hence why it takes so long for me to actually edit anything. Like, I have free hours. Like, for 20 minutes of video, I will sometimes have 3 hours of raw audio to edit down to 20 minutes. So, let's take, I have more of these pipettes. You obviously can't see what I'm doing because I'm on another table, but I'm just fine getting the things. Let's take, actually yeah, let's do this. So, this is some lake water. Now, when I got the water, it didn't have this color. Let's switch over so you can actually see. It didn't have this color. Like, you see, it's kind of white. That's because I put milk in it to give the things in it a bit more, a bit of food, because I went out like hours ago and got it. So to have them a bit of food. They've also been under a lamp to give them some light. So let's see. And I know there's a few water fleas in here, but so maybe we'll get lucky and get one of them. Let's get this. And let's do this. It should be a lot easier. There's a lot of stuff at the bottom of it. Let's try and get that. Well, that didn't. Come on. Quite a lot. Let's see. Get one of the cover slips without breaking it. That broke. They're very thin pieces of glass, so it's hard to. It's sometimes hard if they're stuck. Just like if there's just a tiny bit of moisture in between them, they're just stuck together, and it's easy to accidentally break them. Ah, come on. There we go. Let's put this over here with the broken piece too. And let's put this in under here and see what we can find. And let's switch over. I don't think that was it. Maybe it was. I think this is we have here. Actually, the things we're looking at now are like pretty small. So let's go to 20 times magnification instead. See if we can find anything. This just looks like sand. I don't think I have anything alive in this one, sadly. This looks pretty dead. Which is a bit of a shame. Well, that's a disappointment. Yeah, all of this is just like pretty like small pieces of sand and stuff. There's nothing living in here. That's a clump of dirt. Actually, I think we should move the moss. There's probably a better chance of getting something out of the moss. But I'm gonna just try and take something from the top of this and try that. See if that will work. If that's anything up there. Let's see if we can get one of the water fleas. They're kind of cool. There's nothing on there. The trick is, this is gonna get a bit tricky. Little buggers are fast. Hmm. I think there's too much water. I don't think we're gonna get anything out of this. I mean, that's a bit of disappointment. I probably could find something in it if I waited long enough, but that's we don't want to do that. So let's try the moss instead. So this is just like moss that I put some water in. And there's a good chance that there will be tardigrades in there, and if there is, then I will be in heaven, because I actually did my bachelor's on these little fuckers. So I have a lot to tell about them. We need a pipette. One pipette coming up. See that? Let's see. There's a clump of moss here now. So if we can zoom in on that one, then we should. And we don't really do majors and minors here. You just like do a subject. So I have a bachelor's in biology, and I'm currently working on getting my master's in cell biology. That was, for people watching later, that was for the chat, and there was a question there. Let's zoom in so we can actually see what the fuck we're looking at. So I want to get the big clump here in frame. It's gonna be, if it doesn't really want to cooperate. See if we can find that. There's a piece of moss there. Like a dead piece of something. There's something. Whoa. Look at that. More plant cells. This here you can actually really see the plant cells. Now the question, if you have anything that's actually crawling around here. Is there any little buggers? I wonder what that is. That looks weird. Let's take a look at that. That looks like some kind of algae or something. Possibly some, I'm not sure what that is. I'm gonna guess it's some kind of green algae. That's kind of neat. Let's see what we can find. So the trick here is to look for anything with look for movement, but I don't see any. Like we have to get lucky with this kind of thing. There's a bunch of these algae. Yeah, look. Whoa. Tiny, like those are green algae. Definitely green algae. I think it's mainly them. I don't think there's a whole lot of stuff here, sadly. Huh. It's a long something. Wonder what that thing is. Let's see. We have here. Looks at. I think it's just a piece of stone or something. It just looks like it looked like something. This looks more interesting. It's a bit too big for me to probably get a look at. I think there's something there. Let's take a closer look. Now, obviously, because we have magnified it, not zoomed, we magnified it quite a lot. Like moving around here is not super easy. I think they're just more dirt. Sadly, I think like I should have started with these. All the fun parts were in the other ones, where I actually knew there was something to look at. Well, this is a bit boring. That's what like dirt from a piece of moss looks like under the microscope. I think we need to go back down. Maybe try and go back down to this thing and see if there's anything on it. I think it's mainly just the green algae here. But those are cool too. Well, hey, hello. Hello, we have something. We have something that's very much alive here. Or is that just a movement? No, that thing is very much alive. Look at that. We did find something. Let's see what you are. It's a little wormy thing. Ah, look at it go. I have no idea what that is. It's not a tardigrade, I can tell you that much. Because it doesn't have any legs. We have something else moving past here. I don't think it's looking back at me because I don't think it has eyes. What are you? Look at that. I have no idea what it is, but it looks cool. Well, this isn't a leg sample. This is from some moss I pulled off a tree. It's definitely something it's some kind of animal because it's multicellular. I can tell. I can see that much. It's some kind of worm. Actually, you can see its mouth. I think it might be. Oh, I know what it is. Oh, now that it's folded out, this is a rotifer. You can see if I zoom in here, it has these little. Not zoom in. If I focus, it has these little things at the end that sort of with like hairs that it gets food in with. And then it has you can see the thing in the middle. That's sort of like it pumps the water in and out. So that's what it eats with. So look at that. So it just sits there and sort of tries to catch things. So we did find something. It just had to wait for it to actually unfold its mouth parts to actually be able to see what the fuck it was. Look at that. It's kind of cute. So every time it sort of moves in, I think that's because it catches something. That's at least my guess. That's the wrong way. Oh, come back. Come back, you little bugger. So that's neat. There's the thing about like this sitting here and looking through the microscope. It's not that like there was a reason why I prepared some stuff where I knew there were things to look at, because it does quite often take quite a long time to find anything to actually look at, unless you have a really good sample. Like so. Just the fact that we caught something is pretty cool. Let's see if we can find something else than the rotifer. The scope name, NASA Jet Propulsion Laboratory, blah blah blah. This scope is super old. It's 70 years old. It's produced by a company that doesn't exist anymore called Weishart, and I know I'm mispronouncing that. So it's a Model RC. There's something else moving here. You see that? Let's see. Now the now the trick is to actually be able to focus probably on, because it's moving up and down. What are you? I think we are talking a single salt here. Stop it. You're annoying, you know that? I think it might be a paramecium, which is like a little single cell creature. It's some kind. It's kind of hard to know what it is. So my guess is that it's a paramecium, which is they're small single-celled they're small single-celled organisms. But the thing is, they actually they have small cilia on them. Like they're small, they're not actually hairs, but they sort of look like them. That they move on back and forth, and they actually have like a specific. Even though they're single-celled, they actually kind of have a mouth. They have like a specific spot on the cell surface that they eat from, and a specific spot on the cell surface that they poop out of again. They also have what's called a contractile vacuole, which is like a vacuole is like a little bubble inside the cell, and these when you live in, because there's more inside a cell. Inside a cell there is there's salts usually, because it needs salts to work. But obviously when you live in a lake, the water around you is less salty. So you get osmotic pressure, so you get water that the water tries to flow into you, which first of all it dilutes the salts in you, which makes it harder for you to work. And secondly, you risk rupturing or having some kind of damage from it. So to get rid of the water, they have this contractile vacuole where they pump the water into it, and then when it reaches a certain size, it will attach to the cell membrane, the outer membrane, and fuse with it, and then it will squeeze the vacuole, so it sort of contracts. That's why it's called a contractile vacuole, because it sort of pump the water out and then go back and be filled again. So it's kind of like it's basically their equivalent of a kidney, functionally. It obviously doesn't have anything to do with a kidney in evolutionarily or how it works. It's not as cute as the rotifer because we can't really see it. Yep, parameciums poop. They have a place where they spit out their poop. Well, it's not really poop. It's they will find things like bacteria, and then catch them, eat them in their mouth in quotation marks area, and then they will have another place where they pump the things they don't need. They poop them out. Let's see if we can find something else. The jar, that's the moss. That's where I took the sample from. I don't really see anything. Try and see if we might get lucky and see if there's a tardigrade somewhere. That would be the ultimate prize, just because of how cool and cute looking they are. And yeah, I think tardigrades are cute. They're like little mutant gummy bears. Ooh, I'm not sure what, that's some kind of plant, some kind of plant debris. But it actually looks pretty neat. Let's take a look at that. Ah, look at that. You can see all the, you can see the structure of it with all the little, look at that. You can see all the little hairs on that piece of plant. That's kind of neat. Huh. thought I saw something move up here. What do we have? Lots of small bits and pieces of plant. Well, actually, we've seen a lot of things, and I think I've dogged out enough. The whole point of this hangout was just for me to like dog out and have fun with the microscope. And we've been going for nearly 2 hours now, probably more like an hour and 40 minutes or something like that. So I think I'm gonna head out and not head out. I think I'm gonna stop here and say good night to everyone. It was really, really fun. Yeah, goodbye, and thanks for coming. This was really fun doing this, and even though I had a bit of technical issues. So, see ya.